Dear friends and viewers, in this video session, under physiological aspects of nutrition, we are going to discuss about any vital part of our life, that is nothing but gene. Genes are responsible for migrating the information, genetic information from one generation to another generation. If you want to understand the gene, we have to go for genome maps. So let us see about genome maps. So this session has got its own objectives. Its objectives are just to gain knowledge on gene and the components of the gene and the immunological aspects of the gene. Then we can able to understand the physiological aspects of the gene and the drugs related to that. The outcome of this session is just to recall the functions of the genes and its physiological effects in our body. One can able to discuss the role of various genome maps which is used for our purpose. Friends, in order to understand this genome maps and the importance of genome maps, we have to see the background of central dogma of biology. So the central dogma of biology is very essential to understand the genomics and proteomics. So you can see that in the picture we have our genetic material called DNA. DNA can be duplicated, it's called DNA replication. Then DNA can be converted to another nucleic acid called RNA, and that the process is done by transcription process, so that the RNA which is formed from DNA is messenger RNA, mRNA. Then from mRNA the message can be taken to the next level called the protein level, that is protein language. So this process is called translation. So put together all the three processes, replication, transcription and translation, is called central dogma of biology. So this concept of central dogma of biology, which was proposed by Francis Crick in 1958, this Francis Crick was involved in elucidating the double helical structure of DNA also. So this central dogma describes the flow of genetic information in cells in this route from DNA to RNA, RNA to protein. So DNA is one language, RNA is another language, protein is third language. So that is why the central dogma explains how genotype leads to phenotype. So based on that only we can be able to create a gene map or genomic map. So let us see the genome maps. So in the picture you can see that this is the genome map of an organism or any organism, which is the circular genome map of organism, where you see that all these bands are going to locate the different types of genes and their locations. So this is a typical example for a genome map. So this diagram shows the position of genes and markers. Marker genes are very important genes. They are going to mark the essential of a particular other genes. So these genes, position of the genes and marker genes on the chromosomes. Then this genome map is going to help identify a gene, a particular gene. Suppose here you can see that you have a gene here, you have a gene here, that can be identified. That is, gene location can be identified, and genetic variations can be identified by comparing this genome map with another organism genome map, where the location change can give you genetic variations. Then genome map is very essential for sequencing the genes. Here you can see that sequences are here in this map. Then disease gene identification. Suppose one of the gene here in this map is having some defects, that is a disease gene that can be identified, and the genome assembly can also be identified. Then these gene maps can be used in fields like genetics, and then molecular genetics, medicine, agriculture, and in the field of the biotechnology also. So we have different types of genome maps. So let us see them. First genome map is called a genetic map. This is a map which is based on recombination frequencies of the genes on the chromosomes. The second type of genome map is physical maps. These maps are based on actual DNA base pair distances between the base pairs on the DNA molecule. That is called physical map. And the third type of genome map is called comparative maps. And these maps, they are actually used to compare genomes across species. That is, we have the genome of a monkey and the genome of a human being. That can be compared. This is called the comparative map, which compares genomes of different types of species. Now we'll see all the three types in detail. First one is called the genetic map. Here you can see genetic mapping picture. Here see that this is the genetic map with the sequences, and this is the cytogenetic map of the chromosome, the particular chromosome, and this is the physical map, sequence and numbers, sequencing is there. Then if you take one part of that physical map, you can see enlarge it, you can see the base pairs in the DNA sequence. So this is a typical example of the genetic map. So this map shows the relative positions of genes on a chromosome, this chromosome. Then these maps, genetic maps, they are based on recombination, that is for crossing over, which happens during fertilization. Okay, so this is based on recombination or crossing over frequencies during meiosis. Then these genetic maps, they are going to very helpful for the distance measured, distance between the two genes, measured in a unit called centimorgans, that is called cm. Then these genetic maps are used for higher recombination purposes. That is, what is this recombination? Greater distance between the respective genes. Then these maps are useful for studying inheritance patterns and locating disease genes on the genome. Then these maps are very useful, and they are going to help identify linkage between the traits. Then these gene maps, they are actually the foundation for marker assisted selection in breeding. The second type is physical map. Here you can see that this is human chromosome, human genome, and we are taking one part of this, the dark color one, and we are going to elaborate that. This is the physical map. This is a map of overlapping cosmic clones. They are approximately, this region is approximately 100 kilobase pairs, and you are going to take all these portions. This portion, once if you enlarge it, that is called 5 to 15 kilobase pairs. This is a small clone. Restriction fragment enzyme sites are shown here. This is a physical map. So these kind of physical map, they represent the relative locations of genes on chromosomes. These maps, physical maps, are constructed using recombination frequencies from genetic crosses during meiosis. And these maps, they are useful for identifying linked genes and inherited traits. And these physical maps, they aid in locating genes associated with various genetic diseases. The third type of map is called comparative map. As I said earlier, we can compare the different species. Here in this picture, you can just compare the map of rice, the rice with maize and rice, another variety of rice. This is called comparison. So here, these kind of maps, they compare genome organization across different species. Here you can say rice, third variety, maize, and rice, another sixth variety. So here, these maps, they identify conserved genes and chromosomal regions, that they are called synchrony. Then these maps help trace evolutionary relationship between the organisms. What is the evolutionary relation between rice and this rice, and the evolutionary relation between this rice and maize? These maps are very useful for transferring genetic information between the species, because we are going to compare the genomes of the different species. So now we can conclude like this: genomic maps or genetic maps, they are essential tools in understanding genome structure of various species. They enable identification and location of genes and marker genes on the chromosomes. They support research in genetics and molecular genetics as well as evolution, and especially in disease mapping. Genetic, physical and comparative maps, all the three types of maps what we have seen earlier, they serve complementary roles, because all all the three are interrelated. So all the three can be used to understand the genetic information of various species. And these maps, they are very crucial for advancements in personalized medicine as well as in biotechnology. So genetic maps are very essential for all these. Friends, now this is the assessment time. I'm going to ask questions you can answer. Question 1. Which of the following best describes a genetic map? A. Map based on physical base pair distances. B. A map based on gene expression levels. C. Map based on recombination frequencies. D. Map showing protein-protein interactions. What is the answer? The correct answer is C. Map based on recombination frequencies. Second one. What unit is commonly used in genetic maps? A. Base pairs, that is called BP. B. Centimorgans, cm. C. Nanometers, nm. D. Kilograms, kg. Tell me the answer. Yeah, the answer is B. Centimorgans. Next one. Which type of genomic map is most useful for comparing genes across species? A. Genetic map. B. Physical map. C. Structural map. D. Comparative map. Friends, the answer, yeah, the correct answer is D. Comparative map. These are all the some references. You can refer these links for further information. Thank you very much, friends.